Guess it would help if I turned the microphone on. Maybe a little. Class hasn't started yet. Just getting a quick 15 minutes in of SEM time before your class. Today we're looking at some more pollen. As promised, I collected some strawberry pollen. That's what we're looking at right here. A bunch of wild strawberries growing in my front yard. So these were easy to collect. Well, I suppose everything I collected was pretty easy to collect. Since it's just pollen, I can just grab the flowers and bring them in. And I spent a bit of time prepping them before the stream started. But still pretty easy to manage. So this is the anther for a strawberry flower. If you're not familiar with strawberries, the flowers look like this. So little white flowers with yellow middle parts. And the anther is the little piece that sort of stands up from the surface in the middle, around the fringe, around the outside. So the little yellow bits that are on there that generate the pollen. And when I zoom out, you can see this is one of those pieces. So it redraws it. This structure right here is one of those pieces that generates the pollen. And there's a bunch of them sort of laying around on the surface in here. I just plucked a bunch of them off and also just kind of dabbed the flower on the tape to get some of the pollen to come off. And some of these little pieces came off with it. So you can get a good sense of what the strawberry pollen looks like from these. There's also some other plant cells on here, obviously, which is what these things are. Let me zoom in on this one and see if we can get a nice clean image without it charging. So these have sort of a fingerprint-like ridge on them on the outside, but have 3 long slits in them, sort of like ones that we've seen before. A lot of the pollen we've seen sort of have this structure. They're essentially football-shaped, and then really what we've seen as a primary difference between them is the surface textures are a little different. And I think that actually contributes to the difficulty of identifying pollen in things to like the species level. So you might think, oh, it should be easy to identify different types of plants by their pollen. But as we can see, a lot of the pollen is pretty similar in the SEM, and that means it's pretty similar in the light microscope as well. And then, so there's probably little subtle differences between them that people who are experts in pollen can see, and maybe some things in the light microscope that are a little more obvious, even potentially than they are in the SEM, if that makes any sense. So maybe some aspects of the structure, being able to see through it, that might make it a little bit easier for some aspects, especially if that's the way people are used to looking at them. I don't know. This looks like it's going to be an okay picture. It doesn't look like it's going to charge up. So I'm just going to start us off with something nice. Strawberry pollen. A little bit of strawberry plant. So I think this is the last pollen stream I'm going to do for a little while. Hey, Comrade Bubbles, how's it going? You're at work, checking us out. We're looking at strawberry pollen. So I collected a bunch of different types of flowers today. One of them being these little strawberry flowers from my yard. And then, so we're just taking a look. I take the flower and then I basically pull off the anthers and sometimes a little bit of the petals and just slap them right onto SEM stubs. So, yeah, another pollen day pandemic watch. But I think this will be the last one that I do for a little while. Still slightly congested, but otherwise I'm fine. You know, I don't usually stay sick for long, so I sound a little raspier than usual, I suppose. So you can see the individual cells for the parts of the plant in here. These are long cells, and then the pollen grains are these little football-like things. Here's one on its end, so you can sort of see down the length of it. It looks a little deflated. And then this one, and there's two more that are here as well. And they've got these sort of intermittent ridge-like structures on the surface, sort of whorl patterns. Clearly visible. I think I'm probably going to switch to going back to looking at some more Diatom stuff starting next week. So I've changed my schedule. I won't be streaming on Saturdays anymore for a little while, maybe for a month or so. We'll see. And this is strawberry pollen. Oops. Here's somebody sneaking into my lab. Well, I guess they could be going to the other side. So you can see bits and pieces of the plant parts here, and this component just sort of broke off whenever I dabbed a bit of the flower onto the surface. And the actual anthers that produce the pollen are here. You can see them. They look like little antenna-like structures that stand up from the face of the flower. And you can see that all the pollen basically is really rich in this zone. And then the cells themselves are kind of interesting in here. They have sort of a hexagonal shape to them. And then these ones that are in between where the pollen is being produced in here are a little bit more bulbous. You can see there's a whole mass of pollen right in here in this zone, and that pollen actually moved a little bit. I think it's actually moving. Sometimes the electron beam can, if I focus it on something, will occasionally actually cause it to move. It's not glued down or locked into place very well, which was the case with that piece right there that was in the back. So you can get a nice clean look at all these cells that are right here. You can see how much everything in this area is sort of charging, and that's because it snowed here last night, and the flowers that I picked were still a little wet from the snow. And inside the middle part of the plant here, it's hard to get that dried out. I just let them sit inside for a little while, hoping that they would air dry, but I don't think that they did completely. And you know, like small areas like that are kind of a challenge to get them to actually dry out anyway. So I just plucked this part off with a set of tweezers and stuck it on the carbon tape, which is what you see in the background. This is just the edge of the stub. So now, even though it snowed last night, now it's actually kind of nice out. It's starting to warm back up a little bit. I've been out without my coat for the most part this afternoon, collecting flower parts. So I don't think it's going to stick around for very long, which is nice. These things in here, these cells are actually really interesting. Structurally, they seem very cool to me. Just gonna try to tighten my focus on them a little bit. And then we'll ramp up the image quality. Just snap a picture of that strawberry plant cells right there, so I can come back and see what's going on in chat. What was that on the slideshow that looked like mushrooms popping out of a spore? It was probably mushrooms, comrade. We have put mushrooms on the SEM before. So it's very likely mushrooms. We do that occasionally, whenever we can catch some mushrooms growing. Just finished a math class and then won a game of Wiki. Now you're making a second cup of coffee. All right, Pandemic. I know you love mushrooms. Maybe we'll have a mushroom stream a little bit later in the spring when we can catch some of the mushrooms growing, or maybe I'll just go hunting for them later this week. Fortunately, I'm done with classes already this week, so that's one thing that's really nice. Just by happenstance, by quirk of schedule, someone else is teaching my Friday class, and Thursday class has a quiz and people watching videos outside of class and stuff like that. So my normal really busy Thursday is basically just an open day for me. So hoping to get a bunch done, get some research accomplished maybe the rest of this week, and then next week will also be a pretty light week for me. And then the week after that is finals week, so another light week. So I'm looking at 3 sort of light weeks in a row, and then a whole bunch of grading, which is my least favorite part of being a professor. But it means that I'll have 3 weeks where I can basically get some actual research done, including this week. So I'm looking forward to that. I've got some papers that I need to start working on, or need to finish working on in some cases. I probably just should anticipate getting one of those done, not much more in that sort of a time frame. But we also have a proposal that we need to work on as well. So hopefully we'll be able to make some progress on it. So this strawberry pollen, one of many different plants that I collected pollen from today. And I'm going to move on to the next type of plant that we have here. This is Columbine. So if you're not familiar with Columbine, they look like this. They're actually one of my favorite flowers. This is like a dark Columbine, a purpley colored one. And normally the flower sort of sits kind of face down a little bit. But on the inside they have really bright yellow anthers, and the pollen is very robust in this one because it just opened last couple of days. And again, from my front yard. There's a whole bunch of it that just grows around my yard. It sort of is colonized. I don't think we planted any of it. I think it just sort of colonized itself, which is nice. And you can see all the pollen that it generated here on the stub. And then some of the anthers actually came off, so you can get a good look at all the pollen on the actual anthers right here. The beam is kind of interacting with, again, there's a bit of moisture on the sample, so the beam's kind of jumping around. Because all of the pollen that's here has probably got a little bit of moisture in between it. So we can take a look at that, but probably I can't get a nice high-resolution image of any of the stuff where it's still on the flower part itself, if it starts to charge up like that. I think this is one of the anthers that I plucked off again. And you can see it's just covered with pollen. So, Columbine's very pretty flower, though. And these pollen are the similar shape to the ones that the strawberry pollen were. Again, they're kind of football shaped. And let's get a nice close look at them. I'm going to unblur them here. With the focus height difference. And we'll take a nice close look at them once we get away from the... Oh. It's telling me the filament heating needs to be adjusted, so I'm just going to let it do that for a second. I can come back and see what chat's saying, if anything. Just bubbles. Yeah, Columbine's pretty flower. And sometimes the heating takes a while, a couple of minutes even, to figure itself out. I don't know if anybody out there has really strong allergies, but it's expected that people are probably going to become more allergic to pollen because plants are probably going to start producing more pollen than usual. And the reason for that is at least partly because climate change, global warming issues essentially make it so that it's warmer and wetter in many areas of the country where we live in the U.S. And as a consequence of that, basically, we should expect to see slightly earlier springs, maybe a little bit longer of a spring season. And we can see, actually, now that we've got a nice clean image of these, they aren't exactly the same as the strawberry pollen. Again, the ornamentation on them is pretty different. So these ones have sort of like a stippled or dimple structure on the outside, which makes them look pretty different. You can see that. They look like a plucked turkey or chicken a little bit. You can see that the pollen is all over the place in here. it's also charging up just a little bit, so that contrast and tone change between these parts is partly just a little bit of moisture on the pollen, probably, that's causing that. Suppose at some elevation it won't occur at all, like a little farther away. Let's go see if I zoom around on the slide a little bit. There's so much pollen on it. We ought to be able to find some places where the pollen's not quite so charged up from moisture. There's a big flat area with a whole bunch of pollen right here. It's looking pretty promising. I like it. All right, let's collect that image. Oh, wait. I see. It's jumped the beam intensity back up to 10. I'm going to lower it first. That'll give us a slightly better image and probably even charge a little bit less. But I need to do some brightness contrast adjustments. You had allergies really badly like 2 years ago, and you never really had that before, and then last year stayed inside all year, so you bet you have allergies. Yeah, I usually don't have allergies, but mine is kicked up, and I think that's a consequence of plants producing more pollen. So I think people who didn't previously have allergies, one of the consequences of plants producing more pollen is that you have some sort of threshold, and if you cross it, you're more likely to become allergic to them at a higher concentration. So I noticed it last year, especially because that's going to be a nice, pretty picture. I noticed it last year, especially because we were protected from COVID, so we spent most of our time inside and away from people, and my kid wasn't hanging around other kids as much, and so I just didn't get sick. I don't normally get sick, but like my wife also didn't get sick, and when you're not exposed to people, and you're in just the same 3 people in a little tiny place where you don't interact with a lot of other people except with masks on, and no one's getting sick, you notice when your nose starts to run, when the pollen starts to affect you. So because it's it's separate from a virus or something like that. So it's pretty obvious when you're being affected by allergies in that case, as opposed to something else. So right now, for people who are just joining us, we're looking at pollen from Columbine, which is a type of flower. We looked at a little bit of strawberry pollen at the beginning of the stream, and I collected the flowers, basically put them on stubs, and then put them into the sputter coater and coated them with gold before we started. Reified, do you metallize dielectric sample? I'm not sure what you mean. I've coated these with gold, so they have been sputter coated. Otherwise, the samples would probably be much more charged up than they are currently. And pollen isn't normally metal. It doesn't conduct electricity very well, so that's why we sputter coated them with a metal. In my lab, we almost always use gold to sputter coat stuff because it's highly conductive. And I do have silver. I only really use the silver if we're looking for gold, if we're doing something with metals and minerals and rocks, for example, and trying to do some elemental analysis. But since this is living material, much more frequently for living material, like these sorts of things, I would sputter coat with gold. You can see it's still charging up a little bit down here, this sort of distortion. This is Columbine pollen. Kind of zoom around a little bit. You can see all the pollen on this stub is a highly productive plant. I think the Columbine, like most flowering plants like this that create the flowers, I think their pollen is, well, usually the pollen for wind-blown pollen has sort of a shape that would allow it to be sort of captured by the wind a little bit more readily. And normally when pollen is something that's mostly delivered by insects, it's got a bunch of little sort of burr shapes or spiky shapes. And so if I had to guess, I would guess that this one is probably more of a wind-blown pollen, although I'm not an expert. But normally if it's something that, is meant to be attached to the legs of insects, it sort of has a spiky structure to it that I think allows it to stick to fur a little bit more readily, or to legs of bugs. All right. So unless I can get a picture of one of these anthers without it totally charging up, which maybe I can, give it a shot. So I'm just playing with the focus a little bit. We might do one more where we sort of zoom in on one grain, so you can see the texture a little bit better. Yeah, it's still charging even at 6 in here, so maybe I'll just find a single grain somewhere, and or a small cluster, and something that's not likely to charge up much. Looks like maybe there's some foreign pollen in here. Well, this thing doesn't look like it belongs with the rest of it, but maybe it's just a different shape. Something else in here. There we go. Oh, it's just end-on view. That's what it is. We're seeing it sort of directly in the end-on view of a smaller one. You can see they have sort of 3 little slits in them. So they're football-shaped, and then they have sort of 3 slits in them. And that's a structure that we've seen a lot of pollen have. So I've been looking at pollen for about 2 weeks on the SEM. Just as something to do a little bit different. It's nice to occasionally get away from just looking at diatoms. Try to give people some different views. Some of the pollen views that we've had and some of the plants are pretty spectacular, so they create kind of interesting textures and things. Let's see, I'm going to go ahead and snap this picture. So it's going to take about 3 minutes for it to collect it. And then I can come back and see what chat's doing. Yeah, it's easy to make a really thin layer of gold. That's basically what we have in the sputter coater is just a sort of a gold foil. Still pretty expensive. One of those little gold foil circles is maybe $800, something like that. But for us, the sputter coater, one of those gold plates probably lasts 1.5 years or something. We don't do a lot of sample prep. It's not everyday use, maybe 2 or 3 times a week for a couple of minutes. So that really sort of extends the life of the gold foil for quite a bit. Looks a lot like a plucked chicken to me. With the little pimples on the surface. Yeah, you can see this one's basically just an end-on view of the same grain of pollen with the same structure on the surface of it. And you can see it has 3 sort of furrows or folds that run down the length of it. And you can only see one of them at a time, basically, when they're laying on the surface. So one of these days I'd like to actually do like a stream where I go around and collect something and broadcast the collection process. Problem is that don't get great cell reception in most places where I'm out collecting stuff. So sometimes it's easier just to record a little video of me collecting it, and then I can play the video when something else is going on. I didn't do that this morning. It would have been a pretty short stream. It doesn't take very long for me to collect some flowers from my yard and from around campus. So I didn't think that would be super interesting, although it's nice to get outside. Okay, so this is again Columbine pollen. A Columbine flower from my yard. All right, let's move around. Next up, we have dogwood. So, dogwood, flowering dogwood looks like this. Usually you can tell because the ends of the flower are here. They sort of look like they should, when they connect together, it looks like there's some piece there that's like cut out from the outside edge of the flower. And I actually collected 2 different kinds of dogwood. I think this one is also flowering dogwood. This sort of pink colored one has a similar structure to the leaf, with a thing out here on the outside edge. But it didn't look like the pollen parts had opened up on this one, so I just used the white one, which is a more traditional sort of dogwood color anyway. And here's the dogwood pollen. Again, it has sort of a classic football shape with folds, so not too surprising. I was looking to try to collect a range of things to try to see how different the pollen would be, mostly for fun. I mean, I don't study pollen, but I see pollen occasionally in my work. And it's kind of a cool image. These are the actual cells of one of the leaves of the dogwood flower, or maybe it's actually one of the pieces of the stamen. So just doing a little focusing, and then I'm going to zoom out. Sort of like the flower petal texture in here is kind of cool. I want to get a little bit of that sort of transition from these long stems into this other stuff here. Very cool. Looks like it's going to turn out pretty good. So I'm going to hit that. We'll just collect that image. Someone sounds an octave or two low. Yeah, that's just my sound. Pimpled shell. Yeah, exactly, comrade. The image crispness, yeah. I'm not sure what I have is actually allergies. I think I maybe have a slight cold. But it might also be compounded a little bit by the allergies. But I like to think of it as getting a little bit of revenge because these pollen are being bombarded by electrons. And you can at least see what's attacking you in this case, right? If I called myself pollen attack instead of diatoms attack, I think probably people would think, oh, that actually does happen. Pollen can be pretty vicious, right? Whereas diatoms are benign. Benevolent for the most part, actually. Pollen can have pretty hard effects on people. I occasionally get attacked by pollen. I just love the sort of appearance of the long cells of the flower petals. I got a new lens for my camera this week, and I haven't really had much of a chance to use it because it's been kind of cloudy and rainy and snowy, unusually so for this time of the year. I suppose the rain's normal. But hopefully part of my week or weekend will be spent just goofing around, taking some pictures with it. Part of the reason I wanted it was because it's got a lot longer range than my other lens, which allows me to zoom in even closer on birds and bugs and the moon. So I'm hoping to have some really nice pictures of all of that stuff relatively soon. This is dogwood. Sorry, I keep looking over at the charts so I forget what I was looking at. So I had to make a note, when I put my samples in, which sample was what, so I don't get confused. It's really hard to track sometimes when you have uh, seven random flowers that you stick in the SEM, which one was which, especially if you're not an expert in anything dealing with flowers. But we can see the structure of these. They look a little bit more like raisins than footballs. They're a little more wobbly, wrinkly in here. And you can see the folds very nicely. Again, I think it's 3 folds, 1, 2, and then one behind that we can't see very well. And the surface of this one is also sort of textured with pimple-like structures, but they're not quite the same as the other one. I don't know, I think I kind of like it zoomed in a little bit more like this so we can see our structure. I'm trying to fix my composition. Also, welcome in, Mama Bonbon. Hopefully you're doing well. Actually, when I'm reaching the end of either having a cold or allergies are starting to sort of calm down and my throat is this sort of extra level lower octave, as you say, I kind of like it. I kind of like the extra bass in my speaking voice. A little bit gravelier than normal. Kind of gives me a bit of a growl that I don't normally have. Not normally so growly. But I don't really actually feel bad. I think on Sunday I was pretty tired. And Monday and Tuesday I kind of took naps and slept in a bit. So yep, everything's got sort of its own design, but they sort of follow a similar structure, right? Structural shapes are pretty similar between them, which is kind of interesting. I wouldn't have predicted it, I guess because diatoms are so different from one species and genus from one to the next. Pollen's really similar from one to the next in terms of the base structure. It's like, oh, that's like a pollen grain structure. Sylvia, I think, is the person who infected us all, Mama Bonbon, so she's pretty much over her cold at this point. Or I guess we thought it might have been allergies for her too, but I don't think she's experienced that yet, so that's why we all think it's a cold. It kind of affected everybody in the family a little bit differently, and also like offset. So she's mostly over it and back in school today. And I'm mostly over it. I just have the growly voice. And I think my wife's a little bit tired today. So she's on the other side of it still. Not over the hump yet. But we're all doing fine otherwise. Since I'm completely vaccinated, I don't mind getting a cold, I suppose, because I'm not worried about it being anything else. Definitely like raisin looking. Yeah, it's kind of weird how pollen works. I mean, they're generated in a plant, and then they're either carried by the wind or by an insect, and then think about how random that would be, to just like throw out your sexy bits into the wind and hope it hits another flower of the same kind on a different tree. I'm pretty low probability of that happening. I would think actually for insect transported pollen, that's a much more effective tool, because the insects oftentimes have a preferred plant, and even things like butterflies, for example, they have preferred colored flowers. Like if you take a typical cabbage white, for example, butterfly, which is a pretty good pollinator, they mostly look for purple plants, purple flowers. And so, that's a huge advantage if you think about it, because that insect goes from purple flower to purple flower, dramatically cuts down the possibility of transferring the pollen to the wrong kind of plant, which is just basically the same as losing the pollen for the plant, right? So, that's a much more effective distribution technique to try to get pollen to a target. And especially if the bee or the grasshopper or whatever it is that's bringing it has a preferred target. So the process of pollen distribution and co-evolving with another organism is actually a really good strategy. It has some downfall, which is, of course, if you attach yourself to an insect and your pollen distribution now relies on mostly one insect that is very specialized in delivering your pollen to another type of plant. Something happens to that insect, it also happens to you. So you throw your lot in with another organism, and then you've got to basically suffer whatever that organism suffers. So probably in the short term, it's a great strategy. In the long term, it's not clear to me if that's actually a great strategy. You might be better off being a generalist and hoping you just produce a lot of pollen, and that a bee or something that doesn't have as much discrimination with respect to where it, like what it lands on, is your major pollen distributor, because then you don't have to worry about whether that particular insect is still around or not. So that's kind of an interesting strategy associated with making your pollen, like how it gets distributed, is actually kind of really interesting strategic component. I mean, obviously they don't think about the strategy or the consequences, but just it's the way things happen for the plants. But the evolutionary strategy, there's some like short-term gains, long-term losses potentially for some strategies. But maybe it helps them in the long run, as long as the insect that they've become integrated with or co-evolve with remains successful. This is kind of a nice view of, I think this is again probably the anther, or maybe it's the stigma of the dogwood. I just sort of dabbed a bit of the plant on here, so these are plant cells, these aren't pollen. But you get a nice view of it, so I'm just going to collect that image as well. Oops. Come back to see what chat's been saying. The law of large numbers, yeah. The fact that it evolved that way is mind-boggling. The time it must have taken. Yeah. Butterflies can see ultraviolet markings of flower petals that we can't see. That's true. And so it's probably a combination of color and also stuff that's slightly outside the color spectra for light that allows them to sort of detect it. And also, yeah, hummingbirds are similar. They prefer red flowers. They target specific plants for the sort of sugars that they want, and it's easier for them to distinguish between one species and the next. Yeah, and they also have chemical sensors as well that we don't have. They probably can detect the scent of a flower. I mean, we can detect the scents of flowers, but probably they're more tuned to specific flowers than we are as well. Save the bees. Yeah, of course. Mr. Piece of Work, do you know what type of daisy it was? This isn't a daisy. This is a dogwood, and it's this type of dogwood, a little white flowering dogwood that I collected this particular bit of pollen from. Dogwood is a flowering tree, and I don't know the species name of it, but I could probably find out using iNaturalist or something like that. In the title I have a daisy because I did collect a daisy flower as well. So it's this little purple fringed daisy. I'm not sure exactly what the name of that one is. Again, I could probably figure it out. I'm pretty sure that's a daisy, right? We can go look at the daisy soon. For most of the flowers, I just going by my general knowledge of them. There's one flower that I collected. I don't know what the name of it is. Usually I store some of that information in my wife's head. She's better at knowing the names of flowers than me. This is dogwood plant. Let's move on. Actually, the next stub is daisies and this other flower that I don't remember the name for, so we can jump over and take a look at that. Let's see. These are the bits of the daisy up here that came off when I dabbed it onto the tape, and you can see the little tiny pollen associated with the daisy. Flower parts in here and over here. And then that's just the cell of one of the flower petals on the daisy. So the long purple bits on the outside are these long strands that you see running across the screen. And then I also pulled some of the yellow part out of the middle of this daisy to make sure that I got some of the pollen off of it. And that's all the pollen you see sort of scattered in the background. And then someone can help if you know the answer. Somewhere on this stub, if I zoom out a little bit, I'll probably be able to find it more easily. Oh, here, maybe this is it. Yeah, there's another piece of a different flower that I also stuck on here. These little, this is one of these I didn't know the name of, but I ripped it open. And I did save the flowers, so somebody out there probably knows the name of them. They're like these little bell-shaped flower, purple with like a white fringe around the cone. I don't know what that one's called. Can't remember off the top of my head what the name of that flower is, but that's what this one is. So someone out there probably knows their flowers a little bit better than me. That's what's in here. I had to kind of pull that one open a little bit so we could see the inside part of the flower. But I had extra space on the stub with the daisy, so I just thought, oh, I'll stick it in there. put it on that little extra space because I collected 8 flowers, but my SEM, actually I collected 9, but my SEM only holds 7 stubs. But I put 2 of the magnolias, different types of magnolias, on the same stub. So we can zoom in and look at the a bit of the daisy cell, plant cell structure in here. Oops. Tighten that image. There you go. So that's a really cool view. When we get in close on the SEM, you can really start to see a lot of the underlying cell structure. And they make really interesting patterns sometimes. Struggling to get the image at quite the focal height that I want. Something like this. So you can see the stripes very clearly. And then I'll slow down the beam so we can actually see all the detail a lot better. And I'm going to play with the brightness and contrast a little bit to try to improve the brightness in here because it's a little dark. Whenever I switch from one stub to another, there's a slight difference in the height of the stubs. I mean, you know, micron differences in the height of the stubs. And when I refocus it, often I need to make some adjustments to the brightness and contrast. There we go. That was pretty close anyway. And then I can leave that slowed down, and we can collect this image with a really sharp contrast for these as well. There we go. And let's see what we got. Cornus is the dogwood genus. Yeah. Yeah, we've looked at some stuff from the Aster genus before on the SEM, and the pollen is pretty different. Usually little spiky, much more sort of spiky pollen cells on them. I think I've actually looked that up on I think maybe I've posted on iNaturalist before, so give me a second and I'll see if I can see what it was calling it for the daisy. At this point, I'm wishing I hadn't put so many things onto iNaturalist before. I'm not seeing it. But it just has a light purple fringe around the outside edge and yellow in the middle. And they're very small. Should let you sort it by like if I could pick daisy types, and then you'd probably tell me. Also, I could really quickly see what it thinks this is. One thing I really like about iNaturalist is you can just click on something and take a picture of it, put it on there, Common Grape Hyacinth is the name of that one. Muscari armeniacum. Or Muscari botryoides. But it belongs to the genus Muscari. So, hyacinth. Yeah. Oh, well, that's pretty cool. You're an amateur botanist. I don't study plants. I study algae for the most part. But welcome to the channel. And if you have any insight on any of this, just feel free to spout it out. I'm okay with that. This is Daisy Flower Petal. I know it's sometimes hard to tell from, the webcam view what we're looking at. So, also, we're not going to judge you if you don't know what something is from poor image quality. So feel free to shout stuff out, and then we're in a no judging zone. Let's zoom in and look at some of this pollen. Yeah, this is sort of what I've come to expect from daisies, this sort of spiky pollen. Because again, they probably are mostly insect transported pollen. You can see they're both really little spiky. All the pollen on here is a bunch of little spiky stuff. When I cut open that grape hyacinth thing to put it on here, also it like oozed out sugar. So probably the bees are drawn to that pretty heavily as well. Let's see. I was trying to find one that was a little bit higher density of them, so we could kind of get all the different. Yeah, maybe in here. All the different sort of views of it. There's a whole bunch of different views of the same pollen grains in here, and I kind of like being able to do that. It's pretty tight on the image. I'm going to fix the brightness a bit, and then I think we'll be okay. Take that picture. Hey, P-Chops, how's it going? I need to get P-Chops a shout out. Really, I should just give P-Chops a command. When I go to P-Chops stream, he's got a Diatom command. There's no reason why I shouldn't have a P-Chops command, right? That's just laziness on my part. Alright, so this looks like it's going to turn out pretty good. Speed 7, beam intensity 7, little spiky daisy pollen. I don't know what ragweed looks like, but I thought they're kind of yellow plants, aren't they? And they probably grow in abundance in fields around here, so I could probably get some. Um, I could thank Bubs. Thanks, Bubs. Also, thanks for hanging out with us today, Bubbles. It's nice to visit a bit. I see you post on social media sometimes, and I catch your streams on and off a little bit. I don't always say something. I usually kind of just hang out and lurk, unless I have something funny to add. So, at the base of the petals, there should be a flower structure. Okay. I haven't really studied the morphologies and reproduction schemes of plants, so I'm not sure if the disc flowers in daisies have anthers or not. Yeah, I don't know either. The middle parts have this sort of like little thready structures. When I pulled it apart to put it on the SEM. Here you can see they're sort of cap-shaped, right? So not completely uniformly round. From the top they look kind of that way, but from the side you can kind of see they're cap-shaped. And spiky. And this is the type of pollen that usually attaches itself to insects very readily. So thank you for the follow. Mean Machine Rex. We're closing in on 1,600 followers, so kind of impressive. Managed to collect quite a few people who watch the stream. Daisy Pollen. In the evenings, I sometimes will stream. Yeah, this is sort of these sort of long thread things are what's in the middle of the flower. So when I ripped them apart, this is the part that was in the middle. These are the actual side petals that we looked at. The ones out toward the outside edge. The inside has this sort of a structure, this sort of weird, thready structure. Yeah. Let's roll around and look at the Grape Hyacinth. And this one was a little bit more challenging because it was kind of wet. And also they have these little tiny, really tiny little flowers that are sort of bell-shaped, pointing downward. And I think these are the anthers in here, right here, for those, because they're covered with pollen. And as I mentioned, it was kind of wet when I put it in the SEM, so you can see it jumping around and charging up quite a bit. But you can see all the pollen coating over those surfaces because the anther is where the flower generates pollen in many cases. And these are the tiny, tiny little, these ones are really tiny little pollen grains. They have a different shape than the other ones. These ones are sort of jelly bean shaped. And then on the outside edge, this sort of dimpled or colliculate structure. So that's really cool. Very different than most of the other pollen that we've looked at. So I like that. I was hoping to find sort of a diverse collection of things. And the daisy and this hyacinth have both have very different flower um, pollen sort of structure to them. The shape's different. The number of folds is different. This looks like it just has maybe one fold that goes sort of around the equator or the axis, and then basically it's like a jelly bean or something around that. And again, these were just collected from my backyard this morning. So almost everything in the SEM was collected from my yard. Um, and normally my daughter comes along and pulls up a bunch of the flowers anyways. So yeah, here you go. Very different structure to the pollen on this one. My camera is kind of messed up today. It's chopping my head off. May also be because I'm not sitting up straight. These ones are kind of neat. Kind of reminds me of a pita or a taco, maybe. Yeah, I'm not sure if it's actually pine. Mr. Piece of Work. It could be spruce. I'm not very good with trees. Conifers especially, I struggle with because I don't stop to count the needles or the shape of the needles. But it's right outside of the building, so I can just call it pine for now, and then I'll go look at it later. But I threw out the actual flower piece because pine trees and everything in that group just produce a ton of pollen, and it was like coating the area around my desk where I was prepping the samples. So I was like, yeah, just gonna get rid of that before it destroys everything. Most of the samples managed to get pretty clean, no contamination from one to the next. So I'm kind of happy about that. This is, Grape Hyacinth. Especially considering many of the flowers I brought in a bag, in the same bag, so just in a little Ziploc, basically. They mostly kept to themselves, I think, because they were wet. So I didn't get as much of the normal cross-contamination inside the bag as we probably normally would. But this flower is actually, the pollen is kind of up inside the cone or cup shape, bell shape. end of it, so it didn't seem to have as much of an issue. Okay, so let's see. Maybe do one zoom in here. I don't know if it's going to be a little bit too charged up to actually get the anther here without it sort of going nuts, because it looks like it's really charging. See, when I put the focus the SEM beam into a small area like this, how it jumps, because it's basically just charging up in an area, and then it's causing the image to kind of break. There's a couple of reasons for that. One is that it's way up off of the surface of the stub, and so easier for the material to get charged up. And the other is it's probably a little wet, so the moisture is actually interfering with the beam of electrons coming down. But I can try to focus it nicely on something and then zoom way out. And we could try to just take a picture from a little bit farther out than usual. And maybe it won't be quite so charged up. We distribute the electrons over a wider area, basically. Let's just see how that picture turns out. Maybe it'll charge, maybe it won't. Yeah. Pine pollen's usually just like everywhere. And I find it in my lake, because I mostly do paleoecology and I look at microscopic organisms from lake material. I usually see pollen in it, which is part of the reason why I'm interested in looking at pollen in the SEM, because I come across pollen sometimes in my samples, and it's useful for me to just have a general idea of what different kinds of pollen look like. I can usually get pine, because it looks like Mickey Mouse ears. But the rest of the pollen, I'm just like, I don't know. Really what I should do is try to collect some pollen from aquatic plants. And the lilies haven't started. The aquatic water lilies haven't started blooming yet in my house, but I'll probably do that sometime a little bit later in the summer when they do. Because it would be useful for me to look at those and know what they look like. I don't know what the flowering part of a cattail looks like. I know typhus is a common pollen people find in lake sediments, but I don't know where to collect the. Is it the part that looks like the cattail itself? I don't know what the, where to get the pollen from in that sort of a plant. Oh, this turned out okay. Oh, you can feel it. Is it gritty? I know it's easier to see because it's just like a cloud of yellow around, if you shake a bit of the pollen flower, the plant flower for pine. I saw a picture once of somebody that I don't know if they snapped a tree or if they just, I think, yeah, they'd cut it and it fell. And when it landed, it was just like this pollen cloud came out of a pine tree. And I was like, whoa. But I think I've also seen ones where people just shake the tree somehow, like bump it. And it just, shakes it enough that the pine pollen sort of shakes out of the tree. And it's just like a giant massive cloud of pollen came off of the pine tree, and I was like, yikes. Cattail flowers should be off the plants just before they form the fluff. Okay. I'll see if I can catch some of that in the action. I don't know. total question mark to me whether I'd be able to do that. Okay, we'll zoom out. We're at, we looked at Daisy and we looked at the Grape Hyacinth. And the next thing up is an iris. And I don't know what kind of iris actually. I'm pretty sure it's an iris. Let's see. This was a plant that I collected on campus. So I just was kind of walking around looking for a couple more different types of flowers. And it's this one. So if somebody out there knows their iris, I think it's an iris because it's got 3 different pieces coming out of the center. So like 3-fold symmetry. I suppose it's actually 6-fold symmetry. But there's 3 main flower petals that come out, and then if you pull this back, there's an anther in here. That's this little tongue-shaped thing, I think. And I pulled one of those off, and also this yellow part that's in here. I pulled a little bit of the yellow part of one of these off. This grows on the ground. It's not a flowering tree plant, but an actual flower. So far, Mr. Piece of Work, if you know the answer to what kind of iris that is, or if it's not even an iris, you could tell us. Otherwise, I might resort to using my naturalist again. This is definitely the anther. You can see it's just covered with pollen. This pollen looks a little bit more like fortune cookies. You can see it's reticulated. And there's a ton of it. Let's see if I zoom in on that a little bit more so you can see the reticulation. Nice and sharp. And then zoom out. The brightness is uneven because there's probably some moisture on this as well. As long as it doesn't charge, we can kind of use that to our advantage. But I'm going to try to fix the brightness contrast and see if it evens out the image a little bit. Struggling with it. Yeah. Looks like at least parts of the image should be okay. We'll just see if we can manage to get through the whole thing without charging too badly. This was just a little ground. Like this is the whole size of the flower right here. You know, it was on the ground when I plucked it. So some sort of iris. And you can really see the distinct reticulate structure. And again, it's sort of like a fortune cookie shape, a little bit more like 1 fold, maybe, slightly triangular in cross section. Yeah, I have some really interesting flowers that just show up in our yard. One of them I put into, iNaturalist, and then it came up as a invasive species. I think it's only found in Asia normally. So either the people who lived in the house before us planted it there, or just snuck in somehow. Hey, Pacific Plankton, welcome back. This is some sort of iris. We're looking at the Anther that I pulled out from one of these. It's really hard to get my camera to focus correctly on it, but it's purple, white, and yellow. And that's the whole height of the plant, so it flowers very close to the ground. And these are the pollen that we're looking at right here, individual pollen grains with reticulate structure on the sort of backside. And it looks like they just have one fold right here. The image is charged just a little bit in some of these darker areas, but otherwise it looks good. How'd your class go, Pacific Plankton? See anything good in your sample? This is iris pollen. Perfect. See if some of these clusters are if we can find them off of the Anther. Oh, this is a bit of the actual petal, I believe. Some part I pulled out of the middle of the plant. This is the sort of little yellow stripe bit. So I'm not sure if that yellow stripe is the stamen or if it's some other part of the plant, but you can see the individual cells down here pretty clearly. I think it's the part that lures bugs to land on the plant. Let's see if I can fix this image quality. I'll zoom in a little bit now. There we go. And now back out. And then this flower petal right here, the part of the petal has just a crazy structure. So I'm going to fix the brightness and contrast, and then we're going to take a picture of it. Because anytime there's crazy structures on the screen, I get excited. I like texture, and I really like the texture of plant cells when we're looking at it in the SEM. I think this is going to be good. Set that up to collect the image. Pretty cool. Yeah, it's not bad. Very appealing to the eye. I have this sort of combination of contrast and texture. So the next plants that I had put on here are magnolia, but the magnolia flower petals had all fallen off of um, the plant. And so I just pulled the middle parts out, which I thought might have some of the pollen on them. And I collected two different magnolia flowers. One of them was a saucer magnolia from on campus. Again, all the flower petals had recently fallen off of it. And the other magnolia, I don't know what kind it was. The the flower part that I collected looked a little bit different, so I think it's a different species. But both of them had already lost all their petals, so it was just like the middle part, the anther and stigma components, stamen, I mean, components in the middle that were just sitting there. Oops. This is iris plant petal. Sort of a zoomed out view of the thing we were zoomed in on. And we got a pretty good look at the pollen, I thought. I think that's all that's on here is just one piece with all the pollen and one piece with all the plant cells, but maybe we can zoom in on one of these really closely. Photo from there of the iris pollen. The other one was actually on the anther, and these are loose, or mostly loose anyway. Charging a little bit in the background. As long as it's just a few stripes at the top or the bottom, I don't mind so much because I can crop them. So normally for Wednesdays I don't have time to eat lunch, but I actually ate lunch today, so I'm not starving, and pretty relaxed because I got everything done for tomorrow already, and for Friday, and Saturday, and Sunday, and in fact all the way around. I don't have anything. I mean, I have lectures I have to deliver and things I need to grade, but for the most part, I don't have any work to do. So it's my lectures that I usually have to prepare. I did my last one this week. The other ones are ones I've already given before several times, so I'm already comfortable with them, and I may tweak them a bit this weekend, but it's like an hour or two worth of work for the whole week. Other than that, just delivering lectures and grading stuff. So, light duty, yeah, unusual for me because I'm usually struggling, staying up late and working extra hard, and when it gets towards the end of the semester and students have to work extra hard, I usually get easy mode. So, last 3 weeks, easy mode. So I'm going to replace my normal workload with a different workload. These look nice, though. You didn't say how your class went. Was it good? You see anything good? Next up is the Magnolia. So I was hoping to find some Magnolia pollen. I don't know if there'll be any pollen left on these. Kind of pulled apart the plant a little bit, the middle part, where I assume the pollen would be hiding. And I thought, well, if there's some pollen still on it, we'll see it. If not, I guess we'll just sort of see bits and pieces of the plant. Can't imagine that it could be that close to that much pollen and still have none of it on there. I think this little hairier one was the saucer magnolia. Maybe there's some pollen up here. Looks like maybe a few of these grains did not get discharged. You can see this one has got sort of a little bit more of a rounded shape. This is a bit more jelly bean. I could cheat a bit with these trees because I know that they're magnolia from the flowers, but I knew this was a saucer magnolia. Not because I know a lot about magnolias, but because at the university they put labels on the trees. So a lot of times I can rely on those. I've got a nice arboretum on campus. Most of the trees are labeled with plaques that tell you exactly what the tree is to species. So that's always a positive. See any pollen on this part of the plant. Zoom around a bit and see if we can find any. If we can't, we'll come back to the other piece. Again, very rounded. And dimples on its surface. And also some sort of interesting furrows in here. Let me get really close in. Try to balance the contrast a bit. See how that goes. So as we're waiting for it, might as well wait for it and take a picture at the same time. Yeah, super well defined. Deflated football. I was gonna go with beach ball, but I guess it could be football. This is Magnolia. I think it's a saucer Magnolia from campus. For pandemic, we looked at Iris, Daisy, Grape Hyacinth, Dogwood. I think you were here for Columbine, and strawberry, where we started. Yeah. I'm pretty sure this is the pollen that goes with the plant because it seems to be integrated into the cells, like it didn't get separated from the plant very well. So I guess that's actually the Magnolia pollen. Also, the other ones we looked at were similar. They're sort of round, not as football shape, a little bit more rounded. Yeah. This is probably the anther, so, the part of the plant that's generating the pollen, and so the fact that it's bound to it, it's probably just the way the formation of the pollen occurs. I don't know. But it makes me assume that this is not foreign pollen introduced by wind or something else. By this point, all of the petals had fallen off of the magnolia, and so I suppose it's possible it could be contaminated, but I doubt it. Also, I kind of ripped open the internal part of the flower to put it on the SEM. So it's like, you have the hard, it's like this. This is what it looked like, coming onto the actual SEM, and I kind of ripped off this little bit in here for these, and then there were two different types of magnolia. The other ones I ripped up, on this end, a bit of this piece, which is a little bit like a paintbrush in the inside. Really, I should contact my palynologist friends before I start these streams and let them know that it's going to happen, so they can stop whatever they're doing and spend some time talking about pollen with us. And my friend Chris is sometimes around. I think he catches my SEM images, but hasn't been in on the stream for a while. Maybe I should do a science interview with him. Get him a little bit more integrated. And then when we have questions about pollen, he could help us answer them. Right, let's scan around to the other side of this and take a look at this one and see if we can find any pollen bits hanging out on this one either. Plants just produce so much pollen that it's hard to picture that they wouldn't just have some left on pretty much every surface, but I don't see a lot on these things. It's always possible that you come along a bit late. Maybe it's shaken most of the pollen, or wind's blowing most of the pollen off of it. So it's kind of sticking off the edge of the SEM stub a little bit. A plant cell material, but not a lot of pollen on this part of the magnolias. They look like little tiny Godzillas, wrinkly Godzillas. Or Loch Ness monsters, maybe. It's kind of cool. We'll try to get a picture like this. Some interesting bits of the flower. Very ancient lineage. Yeah, I thought that's what I remembered from magnolias, that they're very old, which is why I thought maybe the pollen would be more interesting on them. The one that we saw just a minute ago is, it's just like a globe basically with dimples. But it's hard to tell because, as I mentioned, most of the flower was gone by this point, so I just kind of plucked off the bits that were left in the middle of it. Maybe most of it's dried up at this point. So either the pollen is inside of the pieces we're looking at, or there's pollen's all fallen off of it by this point. Both are possibilities. You're geeking on the plankton on the background. You mean like outside of my stream? Get your microscope going, just kind of hanging out, listening? Got a bunch of dragon heads on this one. Wormapalooza. Well, on Monday when you were looking at stuff, it was kind of crazy full of worms as well. So, it doesn't surprise me. It's basically the same stuff that we saw. Those little, I don't know, they look kind of like caterpillars to me. Like yellow. I guess the yellow was probably what they'd been eating. Like black stripes on the backs. I mostly took this because I kind of like the texture and the three-dimensional aspect of the flower parts. Just kind of looked like a cool picture to me, so you'll show me later. Okay. Looking forward to it. And you're going to be streaming on Thursday, so, you know, hopefully they'll still be around by Thursday. Given that it's just tomorrow. Zooming around the outside edge of this, trying to see what else we could find. There's just not a lot of pollen on it, actually. And we did find some. So I feel like our mission's at least partially complete for the Magnolia. Hopefully it wasn't foreign. Let's go back to the middle of the slide and see if there's anything there. I think we saw some pollen here when we first started. In retrospect, it might be some of that iris pollen or something else. Hunting for things is the primary activity on the SEM. It's always hide and seek. And found a whole lot else to look for in these slides. Let's move on. And we'll look at the last thing that we had, which I think is a pine or spruce or something like that. I think I glanced at the pollen. It didn't look much like actual pine pollen to me, so maybe it's not pine. But it was all over this middle sample here, so the pine trees are, whatever these are, were in bloom. Look like little peaches. Not like typical pine, at least not the ones that I've seen. So maybe it's a fir, or maybe it's just a different type of pine. The pollen's all over the place on the slide, so that's one nice part about it. I don't have to look too hard to find it. It could also be that it just hasn't opened. Maybe they are pine and they just haven't opened all the way. They are actually quite large. Just running the brightness and contrast for a minute. So we're a little over brightened up in here. It seems like the SEM's actually having a hard time with it. It's got these sort of big side sacs that I think for pine, this is the part that when it opens catches the wind and allows it to be transported. So mostly because their pollen is transmitted by the wind. They sort of have these sort of air bladders or something on the sides, I think. Ved Smocker, welcome in. This is the Scanning Electron Microscope, and we're looking at pollen from a pine tree on campus right now, from the flower of a pine tree. I think it's pine. What's the maximum possible zoom? High. Very high. Hey, Dimas, how's it going? Vedsmocker, we're not anywhere near the maximum resolution on this thing. The magnification currently that we're at is 654 times. And this thing can do magnifications of like 150,000 times. So we're at 654 times. So we're not anywhere near the maximum. We'll zoom in on something here in a second, and I'll give you a sense of exactly how close we can get. We're not even close. This is a resolution and magnification you could get on a light microscope. This is like a 60x objective on a light microscope. We can do way better than that. Is Dell here? Is he hiding? I'm pretty sure this is pine pollen. If you're wondering why is it taking so long to download the image, we're not downloading the image. We're drawing the image. So as it scans across the surface, it's interacting with the substrate, and electrons are being knocked out of the actual material. Electrons are being captured by a secondary detector inside the chamber of the scanning electron microscope. How do you buy an SEM? Well, start by having about $160,000 in your pocket, and then you just talk to the rep, I guess. It's pretty easy if you have the money for it. You can get a desktop one starting at around $60,000 or $65,000. The way that I did it was spent 5 years getting a PhD and another 6 years doing postdoc work, and then got a job as a professor and wrote a grant. That was my solution to getting an SEM. You just have to compete with all the other people. No problem, right? It's easy. And then the university just buys it for you. Piece of cake. I am a professor at Indiana State University. Will be a full professor starting in August. I've been promoted. In fact, Thursday they're doing the recognition ceremony through Zoom this year. So let's zoom in on this guy. Someone was asking, what's our resolution? What's our maximum? And I was telling you we're not anywhere near it. We aren't. Sometimes the material doesn't lend itself to being zoomed in on very well. But let's just zoom in on this and kind of give you an idea of what we can do. At the bottom, down here is a scale bar. That's 5 microns. So, just to give you some simple idea of the kinds of magnification we can get, we're currently looking at just a little piece of this pine pollen, and we're at 15,000 times magnification. So I can do about 10 times the magnification of what we have right now. If I just keep zooming in, for example, at some point what happens is the image will get a little blurry, and even when I have it at best focus height, it will still appear blurry. Right now that's the case because I'm at a working distance of 9 millimeters, and so that means in the camera below me you can see into the chamber what we're looking at, and I can get closer. If I get closer, the image quality will get better when we're this close to the samples. The scale bar down here is 2 microns. So if I want to get really close to something, I have to kind of get the actual instrument closer. So we can do that. Now we're right up on it. And I need to zoom out a little bit so I can figure out where we are. Now the working distance between me and my subject is only about 4.9 millimeters. And that will allow us to get it even closer. So if you pick something over here, like little bits of mold or whatever they are on the surface of the pollen. So, and then we can try to tighten that image up a little bit in here. One of the things is, the closer I get to whatever it is that we're looking at, the the more it matters whether its focal points are perfect or not. And I need to find something that I can get a good, clear image of, which is probably not this pine pollen, because it's sitting on the edge of something. And then play with the stigmation a little bit. I didn't do that today because I knew we were going to kind of be looking at stuff at lower resolution than I needed. But I can play with it a little bit and enhance our image. So that everything we look at is nice and cleanly in focus. We start looking at like the texture on the surface of the pollen grain now. Magnification still only 25,000 times here, but we're kind of in a dark spot. So to get like maximum resolution, I kind of need to be like pollen doesn't just, at some point it just doesn't have a lot of texture anymore, and it makes it hard for us to find something good to focus on. But we can definitely zoom in a lot closer on objects than 25,000 times. That's sort of a starting point for diatoms in many cases. Just I need to play around with the settings a little bit to get it perfect for this. So, and then it takes a little while. So we'll just do one. I want to adjust the brightness because it's probably dark for you guys. It's kind of dark for me. So to answer your question, I've taken pictures at 170, 180,000 times on this instrument. It starts to get a little blurry after about 150,000 times. But, you know, you have to have a subject that's something that has detail at that level, which pollen often doesn't. So, I mean, once you get sort of into the grain of the texture on the surface of it, there's not a whole lot of interesting things that you can see down at that level. If we were looking at diatoms, which we probably will do next time on the stream, we could get really high magnifications without any trouble. So I should probably let that spin past and then slow it when we're looking at the actual sample so I can see the surface. There. I'm going to try to get that just a little bit tighter on the focus. Take a picture. It's looking pretty clean. So this is only 3,000 times magnification, or about 3 times as high as a typical light microscope could do. Here in the kitchen, spilling fermented fruit pulp all over the counter. I was wondering if the SEM can pick up yeast cells. Definitely. You can look at yeast cells in the SEM. I should do that. I should go buy some yeast from the store, some bread yeast. And we could put that on. Yeah, I actually got the news about the promotion, like, I don't know, last semester. So I knew it was going to happen. But, you know, it's still nice. It's like a money promotion, title promotion. It also puts me at the end of my promotions, so I can't get any more promotions at this point. It's just getting old and retiring at this age. Are diatoms what cellite is made of? I don't know what cellite is. Diatomaceous earth. Oh, yeah, yeah. That's what they are. Yeah, sorry. The SEM can do automatic scanning like a moving window, and the examination would be later. Yes, you can buy mods to the software that will reposition the SEM, and it could scan an entire stub, and then you could come back and look at it later. Um, the problem is that you'd have to be kind of zoomed out to do it very effectively, because the depth of focus is very, like when you get down to this range, the side of the pollen grain isn't in focus, like the front is and the side isn't, because the depth of field, the depth of focus on our current picture is only 8 microns, and our pollen grain is, something like 75 microns across, something like that. So, like, once you get around that curve, everything starts to go blurry, and so you have to you have to pick a range where most of the stuff would still be in focus, and then you wouldn't be able to get quite so close to it. But you can have it just scan and take pictures the entire time. Like, I could program it and then come back the next day and have a giant picture. We don't do that. I don't have any purpose for it. I'd rather just do the hunting myself, I guess. Diatoms are definitely algae. Uh, they are a little confusing because they're like plantamoles, right? They're like part plant, part kind of function like an animal in a way. And some of them are even heterotrophic. So, but they are an algae. There are heterotrophic algae, so, um, you know, usually people lump them in with the algae, some sort of heterocont. Um, it's challenging to figure out where to put them exactly, but they're usually called golden algae or golden brown algae. And people move things around occasionally, so who knows. But they're small and they photosynthesize, they're single-celled, so usually we put everything that fits in that category that's single-celled photosynthesizing as an algae, microscopic algae. It's like a whole bunch of them, a whole bunch of little pollen grains on their own. Right here. These will be nice. It'll be a nice collection of things that we can have as an image together. And try to get it into focus sharply here for us. That's close enough for government work. I'm going to just center this a bit. That's the classic pine pollen shape right there, this sort of Mickey Mouse shape, right? When these things start to pop open, I think that's what they mostly start to look like, is these Mickey Mouse shapes. And then the wind catches them, carries them away. So I think these are actually nicely in focus for us. I'm going to slow the speed down to 8 and lower the beam intensity to 6 to get a slightly higher resolution image. And I think this is, we're at the end of our stream because I've run out of things on my stub that we haven't seen already. So, yeah. SARS is very small. And then I'm going to collect this image, and then we'll go find someone to raid while it's collecting it, because this will take 10 minutes to collect this image. I might probably want to adjust the brightness a little bit. And then this is it from the SEM for this week. It's possible that I might be on the light microscope again later this week. We had some really nice stream on Saturday night, and then again, I think it was Saturday night, and then again last night from my light microscope at home. And having a lot of fun looking at stuff with the new microscope settings. So I might go out and try to collect some more samples later this week, and we might do a little bit more light microscope stuff from home, because I have a lighter schedule. I also feel like I can kind of make up some of the night streams. I haven't been doing very many of them this semester. Just been so tired all the time and busy. This looks like it's going to turn out perfectly, so I'm going to just punch that, and then we'll start collecting that image. And I want to say thanks to everybody for coming by and hanging out with us today while we looked at stuff on the scanning electron microscope. This will probably be the last pollen stream for a little while, and we'll probably come back and do some more a little bit later in the summer. I don't know what the streams will be like once the summer undergraduate research program starts, but it might be a little bit more whatever the students are working on, we will stream, and might be a little bit more frequent than usual in the summer. I don't know. I guess we'll find out. I want to say thanks to Calotermes for the follow, for Mean Machine Rex for the follow, Mr. Piece of Work, Swamp Goth for the follow, McCallion from a little bit earlier last night, and Henry Hood from last night. As I mentioned, we're closing in on almost 1,600 followers, so getting close to another milestone. You want to scoop up some pond water and send it to me? You always can, if you feel the need to. I look at pond samples frequently on the light microscope and on the SEM. So I'm gonna let's see who it is I want to figure out who it is I want to raid, because that's how I usually like to end my streams. I don't want to just stop. I want to send you on to somebody else who's also doing something interesting. And I think last time we raided Glorgana, and so this time I think we'll raid Professor Melko, another scientist on Twitch. Just make sure he's not teaching a class right now. Laser repair. Sounds interesting. So we'll pop in and see. We'll pop in and see what Professor Melko is doing. He's part of the Knowledge Fellowship group that supported us early on. And we'll see what Prof. Melko is up to. It's been great having you all visit with me today. Hopefully by next time I'll be completely over my cold, so you won't have to hear me coughing. My voice will be slightly less gravelly than it is today. Yeah, I don't need any rest. I'm fine. At this point, I'm pretty much over it. So I'm going to probably go out and mess around with my camera for the next hour or so. While it's sunny out still, and I can get some images with my new super telephoto lens. We'll see what we can do. Thanks for hanging out, Mr. Piece of Work, and for chipping in on the plant identification stuff. And Pacific Plankton, as always. You should check out other microscope streamers that are on Twitch. Some of my favorite people to watch right there. And we'll catch you next time. Gonna head off for the raid now. We'll see you. Thanks for hanging out.